And the next stage of these 4 stages is the chemical binding or joining of these small molecules into larger macromolecules. So macro simply means huge, like proteins and nucleic acids. Then stage 3, the packaging of these macromolecules into pre-cells. So these are membrane covered droplets, in a way, that have these molecules within them. They're not full functional cells yet, so they're referred to as pre-cells. And the membranes, if you remember that kind of wonderful molecule, the phospholipid molecule that forms the membranes of cells, the phospholipid molecule comes into play here in terms of kind of the self-assembly of those molecules into membranes. And then lastly, the 4th stage for the origin of life is the ability of molecules to self-replicate. If you think of DNA replication, duplication during cell division, whether it's mitosis or meiosis, that's required for the characteristics that we refer to as life, right? The biological characteristics. And this self-replication of the molecules allow for inheritance, meaning passing on of that genetic material from the parental cells into the daughter cells. So there's a well-known experiment back in '53 by Miller and Urey, but Stanley Miller is what your textbook mentions, devised an interesting, fairly simple apparatus that was designed to mimic the early conditions on Earth and to see what would happen potentially under these early Earth conditions. So there was a flask set up, container set up with warm water. Heat was applied to heat up the water, and that was mimicking the early warm primordial seas, causing water vapor, like think clouds. Okay. Then the atmosphere was mixed to mimic the early Earth atmosphere of hydrogen gas, that's the H2, methane gas, the CH4, methane, the NH3 here. So carbon-containing molecules and nitrogen-containing molecules. And then the water vapor, which is H2O, all in the early Earth's atmosphere. And then an electrode was introduced or put into this container mimicking the early atmosphere, and the electrodes would generate little electrical sparks. And that was mimicking lightning that was most likely occurring quite a bit in the early atmosphere. And then they just let this run and let this run. And then the warm air here, the early atmosphere, when it reaches this, what's called a cooling condenser that has cold water moving around on the outside of this tube, well, when the warm air, this little atmosphere, hits the cool sides of the condenser, the atmosphere, gases, condense, form little water droplets, and then anything that is added to those droplets from this environment, this mimicking early Earth's atmosphere, drops down back into this container, mimicking the early oceans, the seas, and then this materials just cycle back and forth, and cycle through this apparatus, and then, so this condenser again causes rain, right? These droplets of water back into the sea, and again evaporation, and it just cycles back and forth and back and forth. Well, Miller let this run for a while, and within days, he noticed that there was material that was streaking along the inside of the flask, and brownish material, and that was collecting into the early sea here, this liquid, and it was cycling back and forth, and more appeared. And again, it only took days for this to accumulate, this brown material. So what this brown material was organic molecules. Carbon-containing and nitrogen-containing molecules. So this famous experiment by Miller helped kind of reinforce the idea that early Earth molecules could appear by these 4 steps. As I was saying there, that Miller experiment back in 1953, showed that the building blocks of amino acids and other nucleotide monomers, some of the simple building blocks of organic materials, could self-assemble in a way with some energy input. That was the little electrodes mimicking electricity and the heat of the warm water. You can get the self-assembly of these macromolecules. So that sets up the stage for then the pre-cells forming, which we'll get to. Okay. All right. So moving along. So the synthesis of these organic compounds. So there's other hypotheses for the origin of these organic molecules on Earth, and they include things like deep ocean vents, which are thermal vents. There's cracks in the crust and water percolates in, and then the hot molten magma, think lava, heats up the water, and you get the bubbling out hot water that's rich with organic molecules. So one hypothesis for the origin of the organic molecules that led to cells, that led to life, is these submerged volcanoes or hydrothermal vents in the ocean, and sometimes they're in the deep ocean. So there are hypotheses that life originated in the deep oceans. There's other hypotheses that these organic molecules reigned and were delivered to early Earth by meteorites and comets, which were much more common in early Earth history. So this sounds kind of very sci-fi, but they've actually have identified simple organic molecules on comets through exploration with robotic explorers that have actually detected organic molecules on comets. So this idea is actually not really so wild. So this abiotic synthesis of the polymers. So how can these early polymers, biological molecules, think like proteins or DNA, how could they have assembled themselves before cells were formed? Once cells are around, then the cells have all the machinery to make the polymers, as we've seen in some of our previous chapters, but abiotic means non-living. So what are the ideas about how these polymers were formed before cellular life appeared? Well, once small organic molecules were present, and there's pretty good evidence that small organic molecules can assemble outside of cells, in other words, abiotically. How are they linked together to form long polymers, like proteins and nucleic acids, without the enzymes or the cellular equipment that are required in cellular production of proteins and nucleic acids? Well, scientists have done or achieved polymerization of monomers, meaning linking monomers together to form polymers, by simple experiments of dripping solutions of organic monomers, like a soup, if you will, onto hot sand, clay, or even rock, and the energy and the surface of the clay or rock material and the heat of it provides the energy and the kind of the environment, the setting where chemical bonding can occur. So in early Earth's history, there was plenty of organic material around, some of these simple building blocks, and plenty of hot clay and sand and rock around. And this is one likely way that the abiotic synthesis of the polymers occurred. So then moving on to the formation of what are called pre-cells. So a key step in the origin of life, because life is cellular, is the isolation and accumulation of organic molecules within a membrane. So here in yellow is the membrane, zooming into a membrane. This blue and red material here is the many molecules of phospholipids that self-assemble into a phospholipid bilayer, which is what a biological membrane is, right? Outside of the membrane, the outside membrane of our cells is a phospholipid bilayer, if you remember that from a previous chapter. And there's other things there like proteins and cholesterol and such. But the formation of a membrane-bound structure with organic molecules within it was a necessary step to develop a more complex living cell. So researchers have demonstrated that these pre-cells can form spontaneously from fatty acids. So you remember the phospholipids have a phosphate group that's hydrophilic, meaning it likes water, attracted to water, aqueous environments. And the hydrophobic tails are fatty acid chains that are hydrophobic, and they repelled by water. So these vesicles can self-assemble. These pre-cells can self-assemble. And then finally, how about the self-replicating molecules? How did that occur? Well, life is defined partly by inheritance, right? One of the major characteristics of life is that the genetic material is inherited from parental cells to the daughter cells. So one hypothesis is that the first genes, sections of repeating nucleic acids on things like a DNA molecule, that the first genes were actually short strands of RNA, which is a nucleic acid, that was actually able to replicate itself without enzymes, because the RNA themselves act as their own enzymes, and these type of small RNA sections or regions or structures are referred to as ribozymes. And this is abiotic self-assembly of an inheritance molecule, in this case RNA. So from there, if that can occur, then there's most likely the ability of DNA to potentially self-assemble as well. And that's an area of active research. So from the chemical evolution to Darwinian evolution. So we've talked about the evolution of the biological molecules that are important for life. So what about the evolution of living organisms? So there is a gap between the pre-cells and even the simplest modern cells, and that gap's pretty enormous. But with millions of years of small incremental change through natural selection, right, aided through mutations, pre-cells and early organic molecules became more and more cell-like through evolutionary change. And again, we're talking millions of years. Eventually, the first living cells were the prokaryotes. So the prokaryotes lived and evolved all alone on Earth for about 2 billion years. If aliens came to Earth back then, things might have seemed pretty boring. Just prokaryotic organisms living in the oceans and in aquatic environments. No trees, no animals, no nothing like that yet. The prokaryotes evolved and flourished on a changing Earth, and they actually helped modify the planet, primarily because of the advent of photosynthesis and then adding oxygen to the atmosphere, allowing eukaryotes to evolve, and especially multicellular organisms. So prokaryotes are found wherever there is life on Earth. The collective biomass is at least 10 times that of all the eukaryotes, which is kind of amazing. If you're able to add up all of the prokaryotic organisms and weigh them, they weigh more than approximately 10 times as much as all the eukaryotes. All the trees, all the plants, all the animals, all the fungus, all the protists. So prokaryotes are very successful. And they thrive in habitats that are too cold or hot or too salty or too acidic or even too basic alkaline for any other eukaryote. So they've been around for a very long time, and they're very successful, and they will most likely be around for a very long time yet.